Bismillahirrahmanirrahim. Assalamu alaikum students. Welcome to PC Science class. I hope you are all fine and fasting in this holy month of Ramadan. Beside fasting, be punctual in your prayers. Kindly do not miss any salah, any namaz. In week 5 we have started the chapter separating mixtures. We will going to continue this chapter, and the subtopics of the chapter which will be discussed today are distillation and chromatography. Objectives of the current lesson are: by the end of the lesson, students will be able to describe how to separate the solvent from the solute in a solid liquid mixture, differentiate between principles of distillation and fractional distillation, identify and separate the different colored components in mixtures such as inks or dyes. Well, you know that a solution consists of two components, solute and the solvent. The techniques or methods which are used for separating mixture, they involve the separation of either the solute from the solution or the solvent from the solution. The three methods which we have discussed in week 5, filtration, evaporation and crystallization, they involve the separation of solute from the solution. Now, how to separate the solvent, which is the liquid component, from the solution? For that, the process is distillation. So distillation is defined, it is the process to separate a solvent from its solution. Now, the procedure of distillation is very simple. The solution is heated. When the solution is heated, its temperature rises to the boiling point of the solvent which is to be separated. So at boiling point, the solvent, it changes into vapors. You must have observed heating of water in your kitchen. When we heat water, after some time, bubbles are formed in the water. When bubbles are formed, we say that the water is boiling and steam is produced above the surface of water. So what is boiling? Boiling is the change of state of a liquid to gas. And any liquid, it boils at a specific temperature. So the temperature at which a liquid boils is said to be the boiling point. Usually water, it boils at 100 degrees Celsius. So in order to convert water into steam, we need to heat the water to the temperature of 100 degrees Celsius. So at 100 degrees Celsius, what happens? Water changes into steam. So to carry out distillation, the solution is heated, and it is heated so that the temperature rises to the boiling point of the solvent, which is to be separated from the solution. Then at boiling point, that solvent changes into vapor. The last step of distillation is to cool down the hot vapor. When it is cooled down, it it changes back to liquid. So this is condensation. What is condensation? Hot vapors on cooling, they change back to liquid, and the liquid which is obtained is said to be distillate. The vapor, when it is condensed into liquid, that condensed liquid is said to be distillate. This diagram shows the experimental arrangement used to carry out distillation. It consists of a round bottom flask containing the solution, and at the mouth of the flask there is a cork fitted, and inside the hole of the cork, thermometer is inserted. The round bottom flask is connected to condenser. Condenser consists of two tubes, inner tube and outer tube. When the solution in the flask, it is heated, at boiling point of the solvent changes into vapor. The vapors, they are hot, they are less dense, so they rise. They rise and they enter into the condenser. They enter into the inner tube of the condenser. Now, the hot vapors, they are needed to be cooled down. So in order to cool down the vapors, water is allowed to flow in the outer tube of the condenser. The outer tube of the condenser, it has two outlets. From one outlet, the water, cold water, it enters, and when it moves around the inner tube, it becomes hot because it absorbs the thermal energy of the hot vapors which are inside the inner tube of the condenser. So it becomes hot, and then that hot water comes out through the second outlet of the outer tube of the condenser. The hot vapor, when it cools down, it changes, it condensed into the liquid, and the liquid is collected in this flask. The liquid which is collected in this flask is said to be distillate. Applications of distillation. It is widely used in perfume industry. Many plants contain fragrant oils which can be used to make perfume. The oil is extracted by heating the plant materials and distilling the vapor. It is also used to obtain pure water from sea water. Here comes the interesting part of the lecture. We are going to see this video which shows practical demonstration of distillation. This flask, it contains colored water. We need to separate pure water from this colored water by the process of distillation. So it is being heated in the flask and it is boiling. You can see that the bubbles are being produced. So when bubbles are produced, the water, it changes into steam, into vapors. The vapors, they are hot, they rise and they enter into the condenser. This is the condenser. You can see that in the inner part, inner tube of the condenser, the vapors, they are changing into droplets of liquid. Those droplets of liquid, they are collected in the second flask. In the outer tube of the container, hot water is flowing around the inner tube, and when it flows in the outer tube, it becomes hot and it goes back from the second outlet. The first outlet and the second outlet of the outer tube of the condenser, they are connected to the rubber tubing. So through the first outlet, this outlet, the cold water enters the outer tube of the condenser, it flows around the inner tube, and then it becomes hot because it absorbs the energy of the hot vapor, thermal energy, and it cools down the vapors. And then it becomes hot and it goes back from the second outlet. You can observe that the vapors, they are changing into droplets of liquid and they are being collected in the second flask. I hope this video is helpful in clearing your concept. Now we will going to do the recap of distillation. Distillation is also used to produce pure water from seawater, and for that seawater will be heated in the flask. At boiling point, the water changes into steam. The steam, it rises and it enters into the condenser. In condenser, the steam, the vapors, the hot vapors, they cool down and they are changed into water. How they are cooled down? Cold water is flowing in the outer tube of the condenser. So that cold water, it cools down the hot vapor, it takes away the thermal energy of the hot vapor, and then it becomes hot. So the hot water, it goes out of the second outlet of the condenser. And then the hot vapor, they condense into liquid, and the liquid, that is the pure water, is collected as distillate in the beaker. Next topic is fractional distillation. Simple distillation is used to separate only one solvent, one liquid component from the solution. But if you have a solution which is basically the mixture of different liquids, in that case, if you want to separate more than one liquid, for that case, fractional distillation will be used. So fractional distillation, it is used to separate more than one liquid component in a mixture of liquids. Fractional distillation is similar to distillation except that it uses a fractionating column or tower to separate different liquid components when they boil off at different temperatures. Applications of fractional distillation: crude oil is separated into different types of oil by fractional distillation. Crude oil basically it consists of different types of oils. It consists of fuel gas, it consists of gasoline, it consists of naphtha, it consists of kerosene, it consists of diesel oil, it consists of fuel oil, it consists of bitumen. So, how to extract, how to separate these oils from the crude oil? The apparatus used over here is a furnace. Inside the furnace there is crude oil, and that furnace it has a connection with fractionating column or tower. This fractionating column or tower it has different outlets. Each outlet is connected to a condenser, and then the condenser is connected to the tank in which the oil is collected. So, what is the procedure? The crude oil is heated in the furnace, and these oils which are present in the crude oil, they will going to change into vapor on their boiling point. Each oil it has its own boiling points. The boiling points of each oil is different. For example, the fuel gas it will going to change into vapor at a temperature of 20 degrees Celsius. So it rises up to the uppermost outlet of the container, outlet of the fractionating column. And then this first outlet it is connected to the condenser. In condenser, what happens that the vapor of the fuel gas they change back to liquid, and then that liquid is collected in a tank. Similarly, gasoline, which is the petrol, it boils at 150 degrees Celsius. So at 150 degrees Celsius, what happens that gasoline it is converted into vapors. Then those vapor they pass through the second outlet of fractionating column. The second outlet it is also connected to a condenser. So what happens to the vapors of the gasoline? When they pass through the condenser, they change back to liquid, and then that liquid gasoline is collected in a tank. Same procedure is followed for the extraction to separate all the other oils present in the crude oil. This diagram shows the recap of fractional distillation. In simple distillation, only a single solvent is separated from the solution. In fractional distillation, it is used to separate more than one solvent liquid component from a solution. For example, we can obtain different types of oil from crude oil. How? Crude oil can be separated into different types of oil by using fractional distillation. For that, the apparatus consists of fractionating column or tower. So the crude oil is heated in a furnace, and then what happens that each solvent which is present in the crude oil is going to change into vapor at its own boiling point, and then each outlet of the fractionating tower or column is connected to the condenser. So when each oil which is present in the crude oil they convert into vapor, they pass through the condenser. In condenser, the vapors they change back to liquid, and then that liquid is collected in the tank. Next topic is chromatography. Chromatography is used to separate and identify the different colored components in dyes or inks. It works on the fact that the different components of a liquid mixture travel at different rates on paper because some components are more attracted to the paper than others. Traveling at different rates, different color components will appear at different locations on the paper. Now we're going to observe how to carry out paper chromatography. The goal of this experiment is to separate different pigments of the ink of this marker. This marker, it has black ink. Do you think that the ink of this marker, it consists of only black color? The answer is no, it consists of more than one color. It is black color of this ink is the combination of different colors, and we can separate those color components by the process of chromatography. For that, we need a black marker and chromatography paper, and a line is drawn at the near end of the chromatography paper. Then it is immersed in a small volume of water in a beaker. It is suspended in a small volume of water in the beaker. What happens that the filter paper or chromatography paper, it absorbs water. The water, it moves up. When it moves up, it takes the color components along with it. You can see that the black ink is separating into different colors. The distance traveled by each color on the paper depends upon how well it dissolves in water. The color component which dissolve more in water, it will going to cover more distance on the paper, and the color component which dissolve less in water, it will going to cover less distance on the paper. So as the water moves up, it takes the color components along with it. You can see that over here the black color, it has been separated into its different color components: yellow, pink, blue, black. So these combination, they form the black ink of the black marker. I hope this video has helped you in better understanding of the topic. Now we're going to discuss application of chromatography. Chromatography is very useful in scientific research because it gives quick result, and it is easy to handle and requires very small amount of the mixture. It is often used to test a substance for purity. It is also widely used in the testing of dyes in the food and in other industries. Scientists, they are able to use ink chromatography to solve crimes by matching documents or stains found at crime scene to the marker or pen that belongs to a suspect. It is also used to analyze urine samples for traces of sugar. In the last, we're going to do the recap of chromatography. Chromatography, it is used to separate and identify different color components in dyes or ink. How to perform ink chromatography? To perform ink chromatography, you put a small dot of ink near the edge of the filter paper, and then that filter paper, it is placed in water. So what is observed that as water moves up the paper strip, it travels upward, it dissolves the mixtures of components, color components, and pull them up the paper. The color components that dissolve most in water, they will move up the paper strip further than the color components that do not dissolve as well. Applications: scientists, they are using ink Chromatography to solve crimes by matching documents or stains found at the crime scene to the marker or the pen that belongs to a suspect. It is also used to test the substance for purity. It is also widely used in the testing of dyes in food and other industries. I hope the lecture is clear to everyone. Take care. Allah Hafiz.